Hello everyone, I am Meryem Uzun-Per from BiletBank R&D Center. BiletBank R&D Center is a division of Akdeniz Pe-Tur. I'm working as R&D project manager in BiletBank. I present my paper called Automated Image Analysis Methodologies to complete Bioink Printability. I completed this work when I was working in Wake Forest University. I coded it in there, but this conference attendance is supported by BiletBank. My paper published in Advanced Engineering Materials Journal in the end of 2020. The other authors are from Biomedical Informatics, Wake Forest University, and Wake Forest Institute for Regenerative Medicine. This regenerative medicine is one of the 3 big regenerative medicine centers in the world. Introduction: the materials used to create artificial organs are known as bioinks. The lack of suitable bioinks in bioprinting is a major limitation in tissue engineering and regenerative medicine. Bioinks are generally consist of a combination of cells, hydrogels, and/or signaling molecules. Cells and signaling molecules dominate the biological behavior of bioinks. The hydrogel typically dominates the flow and mechanical behavior of the bioinks. Water is essential for cells, but reduces printability, and cells are expensive to find and use. In this study, to automatically quantitate the performance of bioinks, methods are proposed, tools are provided using image analysis methods and differential geometry. Several artifact structures are used to test printability of bioinks: a crosshatch to evaluate filament fusion, overhang to test filament collapse, 5-layer tube to evaluate stacked arc accuracy, and novel 4-angled pattern to evaluate turn accuracy. And here are the printing codes of these created structures. Crosshatch is printed like printing 2M letters with 90 degrees. And 4-angled pattern contains turns for different angles. For 5-layer tube, 5 circles are stacked over and over by bioprinter. For overhang structure, a line of bioink is printed on pillar. We give top view of crosshatch, 4-angled pattern, 5-layer tube, and side view of 5-layer tube and overhang structure. And we have calculated 14 different measurements used on these created images using this image. Here, each pore of crosshatch are detected and pore are calculated. PR value is calculated. Angle of deflection is calculated. Angle of corners, width of filament, and standard deviation of this width of filament, and left, right, and straight length of the filament, and thickness, and internal and external radius, and average radius of circles, height, width, maximum width, angle of deflection, spanning success are calculated for overhang structure. Segmentation was used to differentiate the structure from background and determine boundaries of them. Mean shift and thresholding proved the most successful. Mean shift is an iterative procedure in which each data point is shifted to the average of data points in its neighborhood. And in our case, data points are pixels. A bandwidth is set for its neighborhood, and clusters are the structure and the background. In thresholding, the image was converted to grayscale and segmented by global threshold value. In our first data set was created by imaging artifacts for varied in composition with the same printing conditions. The bioinks includes gelingam, gelma, pluronic, alginate, etc. And for different Bioinks, printing conditions were held at constant at 1.4 cubic millimeter per second, flow rate 2.5 millimeter per second, feed rate 0.33 nozzle diameter, and 21 centigrade degree temperature. This data set contains 285 images, 57 images of each case. And second database was created by varying feed rate, flow rate, and nozzle height conditions using a single Bioink in 19 centigrade temperature and 0.33 millimeter nozzle diameter. And Bioink formulation consists of gel gum, gel and glycerol. Various combinations of feed rate, flow rate, and nozzle height were used. This dataset contains 744 images, 186 images of top view of crosshatch 4-angled pattern 5-layer tube and side view of 5-layer tube. This dataset did not contain overhang structure. And here are the selected Bioinks and formulations and printing conditions to report results to cover a wide variety of outputs are given in this table. We will use these conditions to report. And I will both explain measurements and then give the results in the same slides due to time limitation. Here, as I have explained, crosshatches are printed and PR value is calculated by L square over 16A. Here L is perimeter, A is area. Here are we detected each pore and calculated the area. The closer to the one, the squarer the pore. We want to keep these pores as square as possible. So in A, boundaries of crosshatch are indicated by yellow line, and we have smoothed these and indicated them with red lines, and we have calculated area of red lines. B shows segmentation results of top view of crosshatch created by different types of Bioinks and parenting conditions in red, as you may see. White lines shows scale bar of 2 millimeter. And C shows average PR values of pores of crosshatch. Error bars show average negative and positive errors compared to manual calculations. D shows absolute difference of automatic and manual measurements for each turn across Bioinks and conditions expressed as a mean and standard deviation. 4-angled pattern, we have used a uniformity formula, and which is calculated by using length of the left side, length of the right side, and straight length of the continuous edge of the filament, and additional measurement is created, and the other measurements are angles, etc. I have said before. And here in A, you may see template points. We have taken these template points and projected to all of other artifacts, and then we projected these projected points to the boundaries and determined the longest part of this shape, and then calculated the uniformity. And B shows example isosceles triangles created on the skeleton are shown in black, which are used to determine turn points. For each point, 3 points are determined on the line, skeleton first extracted, then 3 points. For each 3 points, we have calculated this formulation, and this triangle, which maximizes these results, are considered as turn point. And we determine all of these turn points and end points, and then we fit lines to each sides of the shape and calculate the angle between these lines. In the magenta lines show segmented, evaluated segment of the filament. Yellow lines show fitted lines to each side of top view of four-angled pattern created by different types of bioinks and printing condition. White lines show a scale bar of 2 millimeter. And in E, automatically computed angle of each turn is shown. And error bars show negative and positive errors compared to manual measurements. In condition 1 here, turn 3 and 4 are not reported since they could not be detected due to deformation of the structures. As you may see, here is a deformed part and we couldn't report it. And in F, absolute difference of automatic and manual measurements for each turn across Bioink and conditions expressed as mean and deviation. And here for 5-layer tube, A shows segmentation results of top view of 5-layer tubes in red, created by different types of bioinks and printing conditions. White lines show a scale bar of 2 millimeter again. And for this condition 5, segmentation of the tube is done incorrect due to reflection on the glass. And correction here is shown in the right side, which is correct, the segment after changing some parameters in code. But we needed to change parameters to create this. And C shows external radius and internal radius values of top tubes in meter. Error bars show negative and positive errors compared to manual measurements. And as you may guess, R is calculated by using area of external and internal areas. And here in a red line, red line shows boundary of side view of the 5-layer tube. Yellow rectangle shows the area which is used to compute the average height by extracting this sloped part of the structure. Magenta stars, as you may see, shows the limit of the 5-layer tube, and we compute max width by using the magenta lines. And here in B shows segmentation results of side view of 5-layer tube created by different types of bioinks and printing conditions instead. White lines show a scale bar of 2 millimeter again. And in C, average height and maximum width of the side tubes in B are shown. Error bars show negative and positive errors compared to manual measurements. And overhang structure, if filament is broken, it's labeled as fail for that gap, for example, in this case. If it's not broken, if it exceeds 4 millimeter, it's labeled as fail again. Here, since the height of the pillars, we take 4 millimeter, and this means this is a failure. And if it passes, for example, in such cases, the angle of deflection of the filament between each pillar is computed. In A, red line shows boundary of overhang structure. Blue stars show left corner of the pillars. Cyan crosses show middle of the filament. And yellow stars here, corner of the right side of the pillars, and red crosses here in the middle of filament at midpoint between pillars. And the angle of deflection are created by calculated by using these points. And this blue line indicates the angle and success of the filament for each gap is reported also above each right corner of the pillar. B shows success and segmentation results in red of side view of overhang structure created by different types of bioinks. Blue lines are used to calculate angle of deflection, and white lines show scale bar of 2 mm. And accuracy of automatic measurements. Manual measurements are taken as ground truth. Accuracy in delineating pores of a crosshatch pattern is 95.8%. Accuracy for each angle of a 4-angle pattern 96.5, 86.0, 80.3, and 80.5%. Accuracy for external and internal radii of a 5-layer tube is 98.9% and 97.9%. Accuracy for the height and width of a 5-layer tube is 90.6% and 99%. Since manual measurements were too difficult to make for overhang structures, manual measurements were not carried out and thus not reported. As a conclusion, we developed automatic image analysis methods to evaluate Bioink printability and propose a new artifact structure for this purpose, 4-angle pattern to measure turn accuracy of Bioinks. Measurements were considerably accurate and showed an excellent ability to differentiate between various printing outcomes. This automation reduces the time and effort required to analyze a structure and also provides a standard set of tools to compare different bioinks. As a future work, we intend to automate detection and cropping image of artifacts. We plan to develop a user-friendly interface in which a user may interactively change parameters for the proposed methodologies in order to obtain a satisfactory segmentation or detection. And here is the citation info. Please cite my paper. Code may be provided upon request. You may take a course from me. Thanks for listening. I may get questions. Thank you. Thank you.